first let's look at the nucleotide's basic structure. So if we zoom it in, we see that the basic nucleotide, which is the building block of DNA or RNA, is really made up of three components. Your phosphate group, we see this group before in phospholipid bilayer. It is exactly the same group as phospholipid. It's called phosphate group because it's the P right at the center here, right? And then there's a sugar here. We call this the pentose sugar because it has 5 carbons in it. So a pentose sugar, pent means 5, O's represents sugar. So a pentose sugar. And the third group that you can find, the nucleotide, that contributes to the significant unique feature of DNA is this nitrogenous base. It's called nitrogenous because you can actually find nitrogen in here, right? So you can see that all this N in these bases. So we have different types of bases and different types of sugar. But the major type of sugar that you're going to learn is the deoxyribose and ribose. You can see that the deoxy has a H group while ribose has a hydroxyl group here. Hydroxyl group. So this gives the differences between DNA and RNA. DNA, remember, DNA stands for deoxyribonucleic acid. Because this nucleic acid is made up of a pentose sugar, this pentose sugar, that has no oxygen in the ribose. So this ribose has an H group instead of a hydroxyl group. So we said that there's no oxygen, so deoxyribose nucleic acid. So it is the main sugar for DNA, while RNA stands for ribonucleic acid. So ribonucleic acid has a group that's hydroxyl group, so the oxygen remains there, so you don't have the deoxy over there, but rather just ribose. So ribonucleic acid is RNA, while deoxyribonucleic acid is DNA, made out of deoxyribose, and RNA is made out of ribose. So you just now learned the 3 different groups that makes up a nucleotide. So let's rephrase it again. A nucleotide has a phosphate group. We usually use circle to represent a phosphate group. Is connected to a pentose sugar. So since it is pentose, we're going to use a pentagon to represent the pentose sugar. And then it is connected to a nitrogenous base. So we usually use a rectangle to represent a nitrogenous base. And there we go. This is what we usually use as a diagram format to represent a nucleotide. A circle, a pentagon, and a rectangle. Phosphate, sugar, and base. So this makes a nucleotide. First, let's learn about the polynucleotide formation. We learned from the previous video, if you want to make a polymer, you need 2 monomers at the very least. When these 2 monomers add up together, it undergoes a process called dehydration synthesis. Dehydration means the removal of water. Removal of water to make something. Synthesis means making. So monomer plus another monomer, we're going to remove 1 water molecules out of that 2 monomer in order to make a connected polymer. With that, we produce a water molecule. And now these 2 monomers are connected by a covalent bond. Here is going to be the same. In order to make a polymer, which is our DNA, we're going to use at least 2 nucleotides here. Here you can see that there's the phosphate group, the ribose sugar, so we're going to make an RNA here because it is ribose here. So 2 ribonucleotide connected, right? When they want to form a covalent bond between these 2 nucleotides, this nucleotide and this nucleotide, they're going to combine together to form a covalent bond, right? This is a covalent bond. And since it is a covalent bond between 2 nucleotides, we're going to give it a unique name, much like the peptide bond or the glycosidic bond. The covalent bond formed between 2 nucleotides is going to be called phosphodiester bond. So if you had 2 nucleotides, we call it a dinucleotide. Basically 2 nucleotides. Di means 2. Nucleotide, the building block of polymer. With that, it's going to create a water molecule. So this water molecule is going to be removed from the 2 nucleotides around here. We take the water out, so now you have a dinucleotide connected by a covalent bond. If we keep repeating this process of connecting the nucleotides together, eventually you're going to form a very long string of polynucleotide, which is a lot of nucleotides connected together.